this video, we'll be studying the phases of meiosis. Now, the easiest way to remember the order in which the phases go is by memorizing the acronym IPMAT, PMAT. Each of the letters in IPMAT should remind you of what the name of that phase is. So the I stands for interphase, the P for prophase 1, the M for metaphase 1, the A for anaphase 1, and the T for telophase 1. So IPMAT will remind you of interphase, prophase, metaphase, anaphase, telophase. Now remember in grade 10, we learned about mitosis, the type of cell division that only happens for the growth and repair of tissue. During mitosis, one cell will divide to form two identical daughter cells. However, when we learn about meiosis, in this type of cell division, during meiosis, 4 daughter cells will be made that only has half of the genetic material. So it's not identical to the original. And meiosis will only happen for the purpose of making gametes, in other words, forming sperm and ovum. So as I said initially, the division of cells during meiosis takes place in a few phases or stages or steps. And we're going to use the acronym IPMAT, PMAT, to memorize and remember these stages. So let's start with the cell at rest. When a cell is not dividing, we say that it is in interphase. Now, interphase is the phase or the stage in between divisions when the cell is just normally, performing its usual functions. But at the end of interphase, the cell has to start preparing for division. During interphase, the genetic material within the nucleus looks like a pot of spaghetti, just a bunch of strands, which we call the chromatin network. At the end of interphase, the cell goes into prophase 1, and the chromatin network actually detangles, shortens, and condenses into these little structures called chromosomes. Initially, when a chromosome is formed, it is a single-stranded chromatid like the one on the left. But soon DNA replication occurs, and that individual chromosome doubles up and now looks like the one on the right-hand side, consisting of two chromatids joined by a centromere in the middle. During prophase 1, a process occurs that we call crossing over. And you might be asked in a test or an exam to draw this process. So this is how it's done. We start with 2 chromosomes. The purple and the green one are actually called a homologue pair or bivalent homologue pair of chromosomes. You'll see that the inside leg of both these chromosomes actually cross over at a certain point, and these ends break off and literally swap around. So the result is a purple chromosome with a little green bit and a green chromosome with a little purple bit. When making this drawing, it's obviously essential that you add some labels. So you should indicate the 2 legs, individual legs of a chromosome is called a chromatid. So here we've got a chromatid on the left. The part in the middle that joins the 2 chromatids together is called a centromere. The point where the 2 legs cross over between the 2 homologue chromosomes is called a chiasma. And then don't forget, obviously, the whole 2 chromatids and a centromere together is called a chromosome. So a question like this could be 4 or 5 marks. Now, at the end of prophase 1, you'll see these little green poles forming at the ends or the poles of the cell, and these red spindle fibers attaches themselves to the centromeres of the chromosome, forming a line in the middle of the cell. Going from left to right, you now see a double line of chromosomes. This double line indicates that we're in metaphase 1, so the M is for middle. Very quickly we see that the chromosomes in the double line start moving apart from one another. Here we go, the top line goes towards the top pole and the bottom line to the bottom. This moving apart is called anaphase 1. As the chromosomes reach the poles of the cell, they will gather together and then form 2 new nuclear membranes, as you can see here. This now, the presence of 2 new nuclear membranes, puts us in telophase 1. This means we've gone through the first division, meiosis 1. So we went through interphase, prophase 1, metaphase 1, anaphase 1, and telophase 1. And again, to remember the order, IPMAT will help. The I for interphase reminds you that it's the stage in between divisions when the cell is at rest. Prophase is when the cell is preparing. So the P for preparing. The moment you see a middle double line, we're in metaphase, and then the A for the chromosomes moving apart until they form 2 new nuclei at telophase. So each letter reminds you not only of the name of that phase, but also what happens during that phase. Okay, so obviously we're not done. Now the cell needs to divide again, otherwise we're not going to result in 4 daughter cells. The 2 new nuclear membrane is going to disappear during prophase 2. Now in metaphase 2 we see a single line forming. Anaphase 2 sees a splitting again, and then 4 new nuclear membranes are going to form, resulting in 4 cells, telophase 2. You'll notice in telophase 2, it seems like there are 4 cells, but they're not distinct from one another. So the absolutely last phase that needs to happen, or step that needs to take place, is called cytokinesis. It's when these 4 subunits actually split and separate completely from one another, and you'll now end up with 4 distinct haploid cells.